really, I'm very happy to introduce you to two friends who will talk to you about the topic we gave us, radical research. The first is Jochen Wittbrod, who shares the same experience. He did research at EMBL, and Thorsten Moos, who is in charge. He's in the Department of Divinities and in charge of ethical questions. So you have an interesting kind of humanities and sciences representation of this place. And I'm very excited about the answers of the questions they will pose to you. So welcome and thank you for joining us.
Great, yeah, thank you.
Also a warm welcome from my side, Jochen Wittbrod. I think what we would like to do, you wanted to present a little kind of stimulative initiation speech, and then we would enter a discussion.
Yeah, sure.
I think we had 3 or 4 topics that we want to go into. Radical research, of course, is the starting point, but the personalized genome, CRISPR-Cas germline editing, and rewriting entire genomes would be one axis, and I think we can't let you go without discussing the mammoth.
Okay.
That was from our side, the ideas of points to discuss after your presentation. And we would allow the participants to ask questions, and I would encourage those who have a question, just type them in the chat. We're gonna see them here, and will be funneled through us, that we have a little bit of opportunity to not only select, but in particular combine if things go along the same line. Okay, that was my introductory words, and without any further delay, I would hand over to you. And well, I'm looking forward to your stimulating introductory presentation.
Okay, so I was told it would be on radical research, so that's what it is. My conflict of interest doesn't really fit on the slides. It fits on this website in the lower right. And I'm very thankful to these organizations and some other individuals and organizations I'll mention. So these are some of the topics that you mentioned. I think there's at least one missing from this that we can easily add. That there's some where we've made a lot of progress already. So gene therapies, we've done a lot on that. And recently they've been applied to vaccines and aging reversal. We've done 2 radical kinds of sequencing, nanopore and in situ sequencing, which were considered ridiculous when we started. Almost everything on this list was at one point impossible or useless or both, usually both. And then another kind of readout is molecular recorders. And I'll mention both what we've done and what is futuristic. And then machine learning for proteins, that was like a 50-year holy grail that suddenly arrived. Same thing, resistance to all viruses. I mean, that still sounds weird, but we just published a preprint that I think proves that, at least for one organism. And then stuff that's more futuristic, and one of which you've mentioned, is GP write, using that sort of whole genome writing level or editing level, multiplex editing for organs, ecosystems. We're getting better at reading connectomes, how the brain is connected with the synapses, but we'd also like to write them. And then finally, we're doing synthetic biology on materials for interstellar. These last ones we won't go into much unless you want to after the slides. So we have these amazing exponentials that are faster than almost any other technology, sometimes 2 or 10-fold per year. We brought the price, for example, of reading and writing DNA down about 20 million-fold, to the point where it's basically, it could be provided, both could be provided free. Another example of lower cost sometimes is, and preventative medicine is vaccines. People don't often think of them, but the top 5 vaccines for COVID-19 were formulated by the same methods as gene therapy. 3 of them were double-stranded DNA in viral capsids, and 2 of them were RNA in lipid nanoparticles. And that, the broad appeal of it brought the price down from about $2.8 million per dose for the rare disease gene therapies to as little as $2 for the pandemic. And smallpox, of course, is $0 and equitably distributed, which is one of the reasons I obsess about the cost, is to make sure that not just the rich benefit, but it is one virus at a time. And so I'll describe in a moment how to get multiple viruses, and also in the very next slide, other common ailments or common disease targets that would justify a low price for gene therapy. And I think that the premier one, in addition to pandemics, is aging reversal. This is going to kill probably 90% of us especially in industrialized nations, which increasing number of us live in cities and such nations, or such nations. So here's an example where we're using multiple genes. It's much easier to do gene therapy than small molecules. We're doing multiple genes at once, and we understand the 10 pathways pretty well, although everything's imperfect, well enough that we can start moving forward. Here's a package of 3 soluble blood-borne engineered genes that we've used in various combinations for these 8 different disease phenotypes in mice, and a subset of them in dogs now, and soon in human clinical trials. This is a database from João Pedro de Magalhães, who was a postdoc in my lab, and Noah Davidson was as well. He was co-author on these 4 papers using AAV gene therapy for all these diseases, and he started Rejuvenate Bio. So that's another example, along with vaccines, where you can get a large market and potentially bring the cost down to $2 a dose. And some of those doses can be lifetime. In terms of reading genomes, we've got nanopores is very synthetic. You've got a pore attached to a helicase or a polymerase. We've worked with 3 different companies. The one that most people have heard of is Oxford Nanopore. These are handheld, long reads, definitely complementary to any other sequencing method. A somewhat related method is molecular transistors. So instead of a DNA going through a pore or going past a pore, this is one where you're getting tunneling between or conductance between a source and a sink, and gated by molecular events, binding or even GATC binding to a polymerase here, bridging the gate and the drain electronic components with alpha helix or DNA helix. And this is some of the raw data from these molecular transistors that we published in PNAS recently. Okay, so that's, but in addition to sort of the nanopores and molecular transistors, we also have in situ sequencing. Now, it turns out all fluorescent sequencing basically is microscopy. And after we developed next-gen fluorescent, we went back and made it into in situ. I mean, it was inspired by in situ, and now it is actually in situ. And we can get as precise as 0.2 nanometers, meaning 2 angstrom. It's a very special case that Ming-Jae Dai did. But getting in the order of 20 nanometers is more routine, up to 40 microns thick. So you get a lot of reads in the Z stack. Here's some of the instruments that we've used. And here's an example of DNA, which benefits from super resolution, meaning better than, say, 250 nanometer diffraction limited down to 20 nanometer, because here you've got 10 kb regions in orange and red holding together a DNA loop. And each of these 10 kb is broken up into maybe 30 of these little three-dimensional coordinate balls. And this is work from Ting Wu's lab, mostly Guy Neer et al. And Boy Nguyen et al. So, to know the motifs where proteins are binding and these loops and so forth requires much better than diffraction limited resolution. So that's DNA. We've done RNA, and proteins are particularly relevant to one of the things on the radical wish list here, which is the connectome. We want to be able to read and write connectomes. And one of the ways that we get a gold standard for all the proteins involved, maybe the RNAs that are non-uniformly distributed, clearly neurons making 10,000 to 10,000 connections per cell is not well approximated by a sphere or a so-called single cell sequencing. And here you can see the 4 proteins all pointing us from presynaptic to postsynaptic and excitatory versus inhibitory. So that's the reading the connectome. We can also store physiological and developmental data as a time series in a linear sequence on the DNA in a variety of ways. We started in 2002, and now there's lots of groups working on all sorts of different ways. I don't want to take too much time on this, but I think it's a radical idea that you can record in DNA. We've done it in vitro for say storing books and movies, but now doing it entirely in vivo, no electronics involved until you want to read it, at least. And we've stored up to 2 terabytes in a body of a mouse. That's only 1/1000th of the body weight is dedicated to storing 2 terabytes. Normally think of terabytes as a fairly big disk drive. And we're moving on to 20 petabytes. We did the 2 terabytes by having 60 sites of CRISPR cutting its own guide RNA and making genome vandalism, which generates random barcodes. So this is like a worst case scenario for off target, but these mice live to adulthood, and every cell in the body has some information about lineage and potentially physiology, as shown by our colleagues at University of California, Irvine, for hypoxia and a small molecule. And then very speculatively, if we can record physiology in every cell of the body, in principle, we could record from auditory and visual cells, and that means that we can now essentially have a movie camera that's the size of the smallest insects, which is this a fraction of a millimeter size fairy fly. There even are retina that fit within a few tens of microns in a protozoan. So the entire retina is within one cell. Another topic which seemed far-fetched when I first was exposed to it almost 50 years ago, when I was starting as a crystallographer, it was how you design proteins efficiently. But we now have this combination of machine learning and mega libraries, which we call ML. Published about 8 papers on this recently, and 4 companies. Nabla Bio does immunoglobulin-like engineering. Manifold does multiplex protein therapeutics, where you can test hundreds, possibly thousands of protein therapeutics simultaneously on one animal. Dinotherapeutics is engineering capsids for gene therapy, and Patch is doing cis regulatory elements in DNA and RNA. We've published on a couple of papers here in Science and Nature Biotech on how we can get tissue specificity and immune evasion by doing these loops of machine learning with up to 1 million different viruses that are designed, not randomly generated. And we've made a big step in this holy grail, which is not based on 3D structure, I'm embarrassed to say, as a crystallographer, but it is based on 1D and functional data, where we can go from a naive model where it's hard to get any, close to 0 with 2 or 3 or 4 mutations out of, say, a region of 28, and we can get to now 28 out of 28 with reasonable double-digit percentages, and 25 out of 28 with close to 100%. The whole library, designed library, works. So we can do things that are much better than CRISPR. I mean, some of these we started on before CRISPR, and many of them are maturing after CRISPR. But these allow putting in really big pieces of DNA, putting in very precise edits. CRISPR is mainly good, or the original double-strand break CRISPR is mainly good at genome vandalism, knocking out genes. But this SSAP with SSB, we found that these work together. Serine integrases, Gabe, Tim, Kale, Tina, and Ben have all contributed to these systems, and they've been applied by these folks in engineering genomes. But both we're doing codons, so there's 64 triplet codons, and we've started by engineering one back in 2011. We started, and now have done worked with 3 and with 7. And the point of this is not just to engineer codons genome-wide, but to make much better use of non-standard amino acids, biocontainment, and multi-virus resistance. And in fact, all virus resistance, you'll see in a minute. Grow Biosciences is a spinoff that Dan Mandel made. That's got a couple of different non-standard amino acids going into clinical trials to stabilize proteins in the serum or to temperature. There are 3 kinds of biocontainment: DNA going in or out of the environment, and the organism itself taking over an environment. And I think we're the only group that's done all 3 at once. And I think it's important, if you're going to do it at all, to do them all at once, so that you don't accidentally create some organism that could compete in the wild. Most lab organisms can't compete in the wild. They're sort of lame and domesticated. But these, if you're literally resistant to all viruses, you might have a pretty big advantage over wild type cells. So here's the evidence that we did that. Back in 2013, we showed that we could get resistance to a couple of viruses. By 2016, we've shown that it was 5 out of 7 viral categories. And you can see these zeros are down from trillions of plaque-forming units per milliliter down to 0 with no recovery of virus. Fast forward from 2013 to this year, actually just a few days ago, we have, July was the preprint. We have 12. We've found there's certain class of viruses that will attack even our best previous strains. So we made a new strain where we swapped, of the 64 codons, we swapped 2 of them from serine, their normal amino acid, to leucine, and then tested them. We tested them with a lot of lab viruses, but we want to test them with wild, sewage, farm material, and so forth, to make sure there weren't. And we could find thousands of viruses that are in this category of thwarting all the best previous strains, and this is resistant to all of them. Now, this is all for E. coli, but that which was the fastest growing organism on the planet until we found Vibrio nitrogens could be harnessed for biotech use, which you can see it's much faster. It's about twice as fast here in minutes of doubling time, 14 minutes. This is the fastest. We've now found examples of both prokaryotic and eukaryotic photosynthetic organisms with mere 90-minute doubling time. Compare that to other photosynthetic organisms like corn, one of the faster farm plants, which is 17,000 minutes rather than 90. So just imagine what this means for for food, materials, and carbon sequestration. And we'll come back to carbon in a moment. So in terms of radical germline editing, we're not doing human germline, but we can affect humans by doing germline editing in animals and then transferring the organs. And this has been done now in a couple of cases. We were invited by the founders of this field and collected all the wish lists of all the people working in the field. It was at least 42 genes in at least one of the strains. We've done this in multiple strains of pigs, both in the US and in China, thanks to Luhan Yang, who's one of the co-inventors of CRISPR and was co-founder of Kihan and eGenesis. And these involve sugars, clotting factors, various immune functions, and we eliminated all the endogenous retroviruses. So this is one of the first edits of a repetitive family, and one of the first complete eliminations of germline viruses. Now, other things we've done with organs. So one way of getting organs that are transplantable, heart, kidney, lungs, and so forth, is in xenotransplantation from animals. The other way is doing it by development in vitro. And we've got, we've done this now for components of the nervous system that produce myelin, the white matter in your brain and the spinal cord that allows signals to go fast. You have demyelinating diseases like multiple sclerosis. So we tested those in preclinical animal models. And then we have two more that are just in bioarchive forms. One for oocyte derivatives, and the other is for granulosa cells that support the oocyte maturation. And then finally, what we're going to use those technologies I just talked about, the multiplex editing, the organ enhancement, and so forth, to help with endangered species and for rewilding, for making species adapted to different environments than they evolved in. So we do this in the context of success stories for rewilding around the world, maybe 1,000 different examples of that, not 1,000 species, but different instances. And probably the most famous one is wolves being rewilded after 70 years of it being missing from Yellowstone. And the wolves restoration impacted the large herbivores, which impacted the trees, which impacted the beavers, and then the rivers and lakes, and then the fish. And so you can see how a keystone species like wolves can have just that one little thing can have ramifying effects. And so we're trying to do the same things with elephants. All elephants are endangered species, and they could be used to restore an environment they used to live in, or their relatives lived in, which is the Arctic. The Arctic has about 20 million square kilometers of carbon-rich soil, much thicker topsoil than anywhere else on the planet, maybe 500 times thicker than some of the rainforests. That's because it freezes and adds layers each year. And we'd like to restore that system to a higher ratio of grass to trees, because grass is better at photosynthesis, it's better at reflecting light and heat, and it's better at allowing the herbivores to pack down the snow. So elephants do like snow, for kind of modest temperature snow, but they make these giant snowballs, and they like knocking down trees. This is a picture of our collaborator Sergei Zimov showing that the karst lakes throughout the Arctic are full of methane. Methane is 80 times worse than carbon dioxide. So there's a very serious problem with 1,400 gigatons at risk compared to the 10 gigatons that the humans produce every year, which is pathetic by comparison of what's at risk. Just melting the Arctic, which is what's happening. Another thing about elephants, part of the reason they're going extinct is there's a killer virus, EEHV. It's a herpes virus. There's also a pandemic virus that affects pigs specifically, which is African swine fever virus. And we just published a paper, bioarchive, on hitting this double-stranded DNA virus with 6 different CRISPRs at once. So this idea of multiplex CRISPR killing of the virus. It's ironic that this is what CRISPR does historically, and it now is there's precious little science and engineering and commerce on using CRISPR to kill double-stranded DNA viruses in humans or animals. And so this is one of the first. It's not like this. It provides herd immunity, where it's hard to go from a virus to go from a genetically engineered virus host pig to another genetically engineered host pig, but it'll go from wild type to genetically engineered and all those sorts. And these are some of the people that have helped on the elephant project. Pleistocene Park. I've been up in Siberia with Sergey and his son, the Zimovs, and Revive and Restore are applying the principles in this project to a variety of conservation projects. Not so much about we de-extinct genes, but it's mostly about helping endangered species. And Ben Lamb has been terrific at fundraising for this. And then finally, there's ionizing radiation. This is not from our lab, but up to 100,000-fold effect of just 4 genes for E. coli, a non-radiation resistant initially. There's this breakthrough star shot where making probes scaled down to 1 gram scale, but they're still flyby. We would like to land something that's 1 billion times smaller, and that's based on synthetic biology. So we're looking at various materials you can make with biology or synthetic biology. Here you can make these little magnetic nanoparticles used for magnetotactic bacteria. I mentioned earlier that you can have an entire retina and cornea, lens, iris, the whole bit inside of a single cell protozoan, and it can detect polarized light of its microbial prey. So, and then you can also make fiber optics. So you can get this refractive index gradient that you expect in human manufactured fiber optics, because it happens naturally in this deep sea sponge spicules. So that's it. Apologize for going quickly, but I wanted to get to the Q&A. Hopefully have a nice long and interesting Q&A. Thank you. Yeah, I'll stop sharing.
Very, very much. I think you gave us a lot to chew on. I want to start with question number 1. What is radical research? Is it radical by being so vast and wide, or is it radical in each of the individual branches? I remember I experienced your radical research in washing southern blots simply with a lot of soap, putting in 7% SDS. Is that an example, or is that just an old thing from ancient times?
Well, so I think radical refers to the individual parts. It is interesting when you get a lot of radical specialties in the same place. Maybe that means something. You're referring to what we call genomic sequencing in a paper in PNAS in 1984. So it is old, but it's also new in the sense that those experiments led from genomic sequencing, which was where we could essentially sequence the whole genome in one gel, but then accessing it required rounds of probing and visualization. Well, that idea of probing and visualization turned into multiplex sequencing, which was in use for about 13 years as part of the Genome Project. And then that turned into next-gen sequencing, because those are rounds of probing and imaging. They don't use the high SDS that you talked about, but did use the concept of probing and imaging, which was quite new in 1984. So yeah, I think that's an example of radical back at the time.
So would you encourage the students just to not just think outside of the box, but really go to the extremes? Or what is a guideline when you're selecting interesting people to collaborate in your lab? Who would you take and who would you not take?
Yeah. So one thing. So one thing is you don't want people who will put their career completely at risk, but you do want people that are flexible of mind. And so I find that people who have already done two different fields, have taken a pretty deep dive, maybe contributed to two different fields, are good choices because it's hard to build an interdisciplinary team with disciplinarians. That is to say, they typically can't speak to each other, and even if you put a big pot of money on the table, they will just take their part and run the other way. But people that you know, if you have two people that speak two languages each, even if they don't have one in common, they at least have the flexibility of mind to find common ground. So I think that, and to protect yourself from, you know, total risk taking, it's good to have one project which is, you know, your dream passionate project, and then another one which was somebody else's dream passionate project, say a decade ago, and is now reaching fruition. And so you can spend 20% of your time on that, helping them get across the goal with low risk to you. But it was still a radical project at one point. It's just you're seeing the end game, and that helps you visualize where you're going.
So to ask a question from another discipline, I think, or maybe part of the radicality of your research might come from that you have a very technical and, or if I may say, engineering approach to the life sciences. It's about reconstructing to understand and constructing to progress, to make advances. I feel quite sympathetic with that approach, since I'm a physicist by first education, and biology was always uncanny to me because it was too complex, too interwoven, too many feedback loops, all that. And this is my question: how do you deal in this engineering approach with the complexity of living systems? How do you take care of that, so to speak?
Yeah, well, so those are all really good points, and I also come from biophysics, so I accepted. So I wasn't blessed with something simple like F equals ma. I was, you know, the things that we would solve would be complex macromolecules, which have a lot of atoms in them, but they're not nearly as complex as a full ecosystem or even a body. And the way you deal with it is you modularize it. I mean, it's not clear that biology has been modularized the ideal way that an engineer would like it, but it's pretty close. You can graft the unlogin, the precursor to an eye into the tail of a developing an amphibian, and it will connect up as if it were an eye, and it will respond to movement. And so it just shows how incredibly modular the whole thing is. You can get two-headed animals and humans, and they act as if that's how they evolved. So they're highly modular, and they're things that you can use in the abstract for engineering. So for example, I mean, smallpox is one of my favorite examples, because we started engineering the smallpox vaccine without any clue whatsoever as to what a virus was or what the immune system was. And those were two fairly key concepts that, but the idea of engineering the vaccines was pretty much the one that worked in the end, was very close to the one we started with. Now we understand the immunology and virology, but we didn't at the time. So it's using modules that work without necessarily understanding the full complexity.
Yeah, I think that I like that approach. I think we're doing it ourselves to some degree. And I think for classical biologists, this may be very disturbing, because basically before you can start engineering, you should have a complete understanding. Is the radical approach to simply ignore that and take what you have and understand the modules and start playing with them?
Roughly, I mean, to say ignore seems a little harsh, but we're willing to ignore it is the point. We're willing to. We're not,
that's
the dedicated to ignoring it. We're willing to, and it's to use, to become familiar with what modules work and for what purpose. So yeah, I think that there are many examples of things where we, for example, when we do differentiation, we've collected a list of all of the potential DNA binding proteins that could regulate differentiation and aging and a variety of other things. And we just see what they do. We ran through all 1,700 of them to see what they do individually. And then we can reverse engineer a little bit of understanding. It goes a long way, where we ask which ones are abundant in which tissues. And then we can come up with a recipe book for making pretty much any tissue we want. And then we can combine those tissues to make organs. So it's all about modules and operational definitions.
So maybe one last general question about the radicality of research before we go into into details. What interests me is what would you say are the driving forces in your research and technology development? Is it the eagerness to know? Is it to solve problems? Is it technology development? Is it economy that drives how do you select topics?
Yeah, so all four of those are excellent, and they do all four of us drive. And if you think of this vast space, I mean, we could do anything, but if you look at the intersection of those four, right, that's a tiny space, and that makes our decision very easy. If we can get an economy of scale, ideally 20 million fold, like the two that I showed, we can have societal impact. We can have some technological progress that might be separate from the economic, which is also technological. And there's some kind of basic science where we drive understanding. We're not forced to understand, but we hope that it'll serendipitously come out of being like you're the first person with a telescope or a microscope. You're going to discover things, whether that was your initial goal or not.
Thanks.
So I think one of the things we had on our list was, well, the genomes, the sequencing, but also the impact of CRISPR-Cas on germline editing and eventually the writing of entire new genomes. I think that for us is interesting because, well, in the interplay between biology and ethics, this is definitely something that is touching a lot of people and causes a lot of, well, responses. Question: Is there a limit in the research you're doing, or would you say the limit is defined by the response of the society?
Well, there are limits, and many of those limits are set by society. I think we embrace the limits. I mean, I think a lot of people think, oh, you're doing radical research, that must mean that you're some renegade that doesn't like rules. On the contrary, I think I'm probably one of the premier advocates of and enthusiasts for the Food and Drug Administration in the United States, and it's called the EMA in Europe, CFDA in China, and so forth. That these organizations that look for safety and efficacy of drugs, we respect and fund. The EPA, Environmental Protection Agency, and their equivalents worldwide, and U.S. Department of Agriculture, USDA. These are watchdog organizations that I think we owe a great debt of gratitude to. They protect us against, like the FDA protected us against, for things like Vioxx, hormone replacement therapy, thalidomide, and the list goes on. We don't want to push the limits of that. So with the case of germline, I think that's, I'm not I think it's a case where we've kind of exaggerated the risks relative to other things. It's not that there's no risk to germline, it's that there are other versions, in particular somatic editing, or just somatic gene therapy more broadly. It's very powerful, and it's more likely to impact us than the germline is. The germline is restricted to a tiny fraction of people that are being born compared to the 8 billion of us that are already born. And so it's much more likely that if we get a gene therapy that works in adults, it's going to spread. If we're worried about augmentation, it's more likely it's going to spread in an adult population. For example, if we want to fight neurodegenerative diseases like Alzheimer's, we might come up with something that's mildly enhancing for someone who's cognitively impaired, but that could be used off-label on people that are not impaired. And that will happen a lot faster than if you have to go through a 30-year debugging cycle where you go germline, see if they're cognitively enhanced. If not, go back to square one, do another 30-year trial. It's you can do, in principle, much faster cycles in adults. So I think the germline has been overemphasized. Not that it's unworthy at all, it's just that there are other things that are more promising and more threatening.
We've seen the promise. You presented that very clearly, but you mentioned the threat. Where do you see the threat?
Well, so one of the things is inequitable distribution. That's why I've emphasized low cost and educational efforts and other outreach that might be just ways of getting people to pay attention to the revolution. So it does no good to have something that's low cost if nobody knows about it or nobody trusts it. So that's one thing on the rich get richer kind of risk. There's a risk that you could enable new categories of warfare. I mean, in principle, you can harness all kinds of things. You can harness a jumbo jet full of fuel to do a great deal of damage by terrorists. But I think a bigger threat in terms of warfare is probably making engineered viruses, which doesn't require germline or CRISPR or any of that. And I think it deserves a great deal of attention to preventing it.
You talked about the watchdog organizations like FDA and the others. I would like to know, or let me start, at the University of Heidelberg, we recently started a project, Bioethics in the Lab. We're trying to involve students, young scientists, experienced scientists, and we're discussing latest developments in life sciences. The idea is to make sciences from beginning with the students, to make them a bit more capable to speak about the concerns they have, the concerns others have. What would you say is the responsibility of scientists, apart from the watchdog organizations, the responsibility of scientists, especially young scientists who work in these emerging fields?
Yeah, I think that's really important. There's a lot of emphasis. I mean, in almost every profession, there's a long education that doesn't always historically involve ethics. It's like you're going to spend years doing this. Why not have some of those years spent on ethics? I teach an ethics course, which is actually required of the students, it's not required of the faculty. In fact, it's fairly rare for faculty to teach it, but which is called Responsible Conduct of Science. It's required by the NIH. It touches on some of these topics. Another thing that helps with the younger, even younger than this NIH requirement, is the iGEM, which is International Genetically Engineered Machines competition, which is worldwide now, involving hundreds of universities all over the world. And from the very beginning, we had a challenge as to what would the reward, what would we be rewarding? What would be the contest in iGEM? What would be the goal? And we looked off to the side, and there was a contest that had been in play for a few years, which was called Robot Wars, where all the students would build robots that would win by destroying the other robots. And we felt that was a very bad idea for synthetic biology, and we stayed as far from that as possible. Instead, we required, in order to win the prizes, you had to have a component which is called human practices, or an ethics component, where you would talk about safety and education and so forth as part of your. You might be that you were creating some new life form, but you were also had to talk about safety and policy and education. But you have to start early. You have to develop it so that it's a knee-jerk reaction. You immediately, as soon as you think of something new, some new technology, you immediately say, what can go wrong? You invite lots of other people to think of what they think could go wrong, and then start protecting before you even develop it.
Start early.
We have a question, maybe that relates to start early. Question from the audience, Sebastian Knobloch, and he's interested in whether the genome sequencing eventually will help to improve matchmaking.
Well, it actually has been improving matchmaking since the mid-'80s, thanks to a rabbi who had 4 of his first children were afflicted with Tay-Sachs disease, which is a very serious recessive disease, kills kids around 4 years old. He decided that they needed a better matchmaking system, at least for his community, and developed Doriya Shurim, which is now, I think, impacting maybe millions of people worldwide, mostly in Ashkenazi Jewish community, but the principles are applicable to everybody on the planet. There's nobody that's at significantly lower risk than the Ashkenazis are, because everybody is at risk until you learn your genome. So ideally, this would be applied pre-conception, premaritally. So you lower the consequences of the information. You even almost eliminate the consequences of false positives, because it's not affecting your health as such. You don't have to go and get a surgery. It's just affecting your matchmaking, which is fairly innocuous, since there are usually thousands of people that you could find a match with. Now, that may not have been the intention of your question. That you might have been talked about who will be compatible with whom, but I can say that whether you're compatible when you start out, if you have a severely diseased family, it puts a lot of stress even on the best matches. So I think a lot of people can get along pretty well as long as there's not a lot of financial and health stresses on them.
We have still some other questions in the chat. One is about understanding viruses like influenza, coronavirus, and the question is, they arise substantial social concerns. Do you think the society is ready to embrace radical research if it comes to genetic modification on viruses?
So I think we are ready for radical research, period. Not necessarily every kind of radical research, but I would argue that using gene therapy for vaccines, and I mentioned that the top 5 vaccines were essentially gene therapies, that was a radical move. And in fact, it was embraced very quickly. I think a record was set for the fastest FDA approval in history. It was, it was 1 year rather than the usual 10-year approval. And some people said that was rushed, but I think I'm not a fan of rushing the FDA, but I think in that particular case, there was benefits greatly outweighed the risks, and the risks at this point, looking back on it, were really quite low. But what important is what the risks are perceived going forward. So I think that's an example of radical. Now, that's not radically doing germline editing or radically changing viruses and then releasing a bunch of viruses into the environment to fight the bad viruses. I think those are radical in a direction that we may not be ready for. But I think we're ready for the idea of using gene therapy on billions of people, which is pretty radical. So I think you have to be careful when you say radical. It doesn't mean every possible radical idea. It's carefully selected ones.
Okay, there is a new question that just came up, and that is human trials and experimentation as a long and difficult process. How can radical research improve this step to overcome and make things more easy for research and implementation? Okay, is radical research improving human approaches eventually?
Yeah. So I think that the question is about long and difficult process of doing the R&D for clinical trial and final approval. Like I said, it usually takes 10 years, and that's an appropriate amount of time. You don't want to necessarily speed that up, but you do want to increase its success rate, because if each one of those costs $100 million, then it might average $500 million to $1 billion to get one success out of 10. So you might want to speed that up a little bit, but you mainly want it to success rate to get higher. And one of the things I mentioned in the brief talk was this manifold bio, which is figured out a way to test multiple therapeutics simultaneously by using barcoding. You can test for effects on toxicity and efficacy on a kind of a body-wide basis and multiple protein drugs simultaneously, maybe thousands at once. And so what used to be a very expensive task, where you might need thousands of animals or humans, you can now do in smaller number, maybe 1,000 times fewer to get the same statistical significance. A second thing that could really help is to make a more realistic model than an animal model. And so we're now making human organoids and increasingly organs that are more and more realistic, closer to the complex physiological systems that involve multiple organs. And then so you can do safety and efficacy on something that is human. So certain drugs succeed in the animal preclinical trials, but then fail in humans. And that's can be, we don't want that to happen.
You would see the future in organoid or organ-based drug testing systems that directly use human material.
I think that yes, I think that either better humanized animals or completely human organ systems. The tricky part about organ systems, they have to get much better than what we have now, but it's going fast. Ideally, there's interactions of the brain, the neuroendocrine system, the immunology, lungs, liver, all these things interact in processing pathogens and toxins. So, but we're getting there. So I think that humanized animals, human organs in vitro systems, and this multiplex testing system that Manifold uses.
We have another very interesting question, which is again to the topic on the topic of complexity that we already discussed. It's by a biology student, I don't know, but it asks: is there a shift of competencies in biology from classical biological competencies towards engineering, towards physics, towards chemistry? And what are the roles and challenges for a new generation of biologists in this field, in this emerging and shifting field? What do we have to know?
Well, sort of from my point of view, which is, I don't mean to say that I'm, have interviewed all the next generation, but I think we are seeing a larger input from physicists, chemists, and especially interdisciplinary people, computational biologists. I gave some examples like the machine learning and mega libraries. That's a big input from engineering, computer science. Now, it's not to say we haven't had previous revolutions that involved outsiders. The dawn of molecular biology was arguably by a lot of physicists that brought knowledge of radioisotopes, stable isotopes, instrumentation and so forth. But every time we've seen that influx of multidisciplinarity, we've seen a revolution. I think there's also a need for people who can wrap their heads around complex biological systems, and that's much closer to the classical biologists. So I think we need all of the above. We need teamwork, and we need people who can work with people and all that sort of thing.
So that, in theory, should require that we're massively rewriting our curriculum, right? And maybe offer to study rather radical research than classical biology.
All of the above, yes. And the problem is, how do you fit all of the above into a reasonable education time? The similar things have been faced in the past. How do you get an MD and a PhD? How do you, get both computer science and biology to become a computational biologist? But time and again, we figure out how to do it. Part of it is by modularizing the key parts of the discipline you actually need.
I guess in the end, we have to modularize the teaching as much as modules are kind of repurposed in the research approaches you're taking.
Yeah, and don't be too insistent about learning everything the way that your father did. It's like there are some things that will have to be left behind. Before recombinant DNA, there was hybridization kinetics and sedimentation and all sorts of biophysical chemistry that I learned that it didn't feel I needed to teach to the next generation.
I guess what we see here is that biosciences in particular, they are pretty modern. And I think we teach on CRISPR, not in the first year, but definitely in the second year. Whereas my son studied physics, and he said, well, the first new thing we learned was at the beginning of my PhD. And I think to that end, biology is pretty dynamic already.
Yeah.
So for a student who doesn't know what to do, what would be your recommendation? How should they start? What is their thing? Well, I have things that I would say, but I'm curious what you would say.
Yeah, I think the first thing is, I don't think there's a one-size-fits-all answer. So you should somewhat march to the beat of your drummer, not everybody else's. But also, I think that you should, I've mentioned the interdisciplinary theme before. If you can get some interdisciplinary training, that would be good from the get-go. But do it by two specialties necessarily, rather than trying to cover everything. And then finally, or maybe initially, learn by doing. If you actually try to make something rather than understand, as your first exercise is to make something, you get a better feeling for what's important and what isn't, and you get the kind of the satisfaction and the positive reinforcement of making something. So I would say try to do an experiment or build something as an instrument or a new cell as soon as possible.
So that and the iGEMs is a perfect starting point.
Mm-hmm. Yeah.
So what do you say? What does that mean for the general audience who's not educated in one of the life sciences as citizens? If we looked at your talk, and some of us might feel a bit uneasy about all that is happening, what I barely understand, what would you say is the role of citizens in this field? How should we as non-biologists educate ourselves in this field to judge all that what happens? Or should we keep quiet and leave it to the FDA and the watchdog?
No, I'm a big believer in citizen science. There are a number of things that you can do. I mean, one is to participate. I've been a guinea pig in a variety of medical research and other research topics over my lifetime, starting when I was a child, actually. And I think that's one way that you can, you again, learn by doing, learning by participating. And if the scientists running the project aren't telling you what's going on to your satisfaction, to keep asking. The other thing is, you don't need to understand all of the, or even a fraction of the theory behind it. What you need to focus on is the outcomes. How do they show safety? How do they, how do you compare a new drug with an old drug, or with a placebo, understand that kind of how to and what comes, so for example, I think citizens participate a lot in the science of cell phones, right? But they don't necessarily understand how many GPS satellites there are, how many you need to get a signal, how atomic clocks are used to get really precise. Is it required for GPS to work? But they can still ask, does it fail? What happens when the GPS fails? What happens when when a hacker gets in, and so forth? So focus on the outcomes more than the mechanism, and then the outcomes may lead you to the mechanisms, but it'll lead you to in a prioritized manner where the failures are the things you want to understand rather than understanding everything.
When I look into the chat, we get into an interesting spin. Apparently, you've really intrigued people, and they want to know what they have to do to become like you. What is it? How can they? What should they do? Or maybe what they shouldn't do could be more important, because I don't believe that there is a recipe to become rich and famous. But you can probably avoid to become, well, poor.
Well, yeah, I think you're hit the nail on the head. There are things to avoid. You definitely don't want to take my path. Mine was a rocky path. I had to repeat 9th grade. I had to repeat part of graduate school. I flunked out of graduate school. Lost my major grant just before I came up for tenure, which is usually fatal. So I don't recommend any of that. But more importantly, you should, embrace all the ethics that we've talked about so far. You should be sort of a compulsive truth teller and truth seeker. I think if you that will help you no matter which direction you go, hopefully. And follow your passions. That motivation is almost everything. I think it's 99% motivation and 1% inspiration, to paraphrase Edison. So find something that where your motivations somewhat and likely align with society's motivations. Yeah.
That's exactly it. Yeah. Thorsten? No. I think we still have one question from someone who's concerned that maybe the policymakers and funding bodies may not be really good at catching up with the progress. Do you see that they improve and that we can, see a better, let's say, company by those in the future?
Yeah, I actually don't think it's that hard to catch up, but I do agree with the basic sentiment that our policymakers should show more interest. The people who catch up easily are the patent clerks and the FDA. So the FDA managers, or the governmental forces that help establish safety and efficacy, they're actually bench scientists. They have, almost all of them have active labs, which I greatly enjoyed talking to them. I've been to give lectures there and to get advice there many times, and they're very knowledgeable. So they keep up. They can keep up. The people who can't keep up are some of the top leaders, and that depends on the country. So I would say, for example, in China, they take science very seriously. I've heard up to 80% of their top politicians have some kind of education in science and engineering, and that's because they were good politicians, not because they failed at science and engineering. And that shows because, for example, there was 400 times fewer deaths, by my reckoning, in China than in the United States, even though they have a larger population, and they had less time to prepare for the pandemic. So a factor of 400 times death rate is an enormous price to pay for ignorance. And I apologize for my country's ignorance.
Okay. So another question that goes still a bit further from the chat. Do you think it's necessary to be a geneticist and know a lot about science in order to found a company which key technologies about based on knowledge about the genome? So we have also economists watching our talk maybe. How much do I have to know about biology for being economically successful?
Yeah, so I think that a typical startup company needs a small village. I mean, it needs people that have business administration skills, that has economic insight, marketing insight. It needs physicists and instrumentation people typically because there are instruments involved, quality control experts, and so on. So if you fit in almost any category of modern industry, then you can help with biotech and genomics industry. I think it helps to again focus on the outcomes. If you're interested in genetics, ask just a tiny sliver of genetics that's relevant to your proposed company, or maybe a few others that are in your immediately competing technologies. I don't necessarily mean economically competing, but other possible ways of doing it. You want to make sure you're not blindsided by the next wave of technology. So you do need to know a little bit, but mainly you're going to depend on your team. You just want to know enough so you can communicate with them. And that's not very much, frankly, to get started.
I promised that we would come back to the mammoth, and you've indicated at the end of your presentation that this is ecologically highly relevant to bring them back in order to contain the methane. Technically, how would you really progress from recent species to the extinct mammoth?
Well, so our emphasis is on the saving the endangered species and restoring the environment, not necessarily bringing back a specific species. So we can bring back extinct genes, and we've done that already. And we can put in multiple genetic changes in an organism and make them so that they can now breed. You don't need to do the heavy molecular biology and cloning every generation. You just have to get it going. And we've shown that we can do that in pigs, as I mentioned. But we need to come up with ways that we can get scale up so that we don't, in a way that doesn't interfere with the reproduction of the endangered species. And we're trying to do that by this organogenesis that I talked about earlier, but focused on reproductive organ systems. And that's moving along very quickly, especially in mice and in humans because of in vitro fertilization. But the elephants will catch up on that. Maybe there's some other aspect of the question that you wanted to emphasize.
No, I think It pretty much illustrates it. In the end, with an animal of a generation time of 30 years or so, I would have more a personal question, because you're starting an experiment that you're not going to see in the end. Well, we all do that when we start reproducing, but how does that feel? You won't be able to really see the ultimate effect of what you start. The promised land.
I think we might see okay, for a number of reasons. So for example, when I started my journey to reduce the cost of sequencing, if you took, it seemed unlikely that we would be able to keep up with the amazing record of electronics industry. Electronics was improving at about 1.5-fold per year, and it had been doing that for decades. They knew how to do it. They had a roadmap. They were skilled at engineering. Biologists were very poor at engineering when I started in this field. And so if you extrapolated that very aggressive electronics curve called Moore's Law out, it would have taken 6 decades to get to an affordable genome. Instead, it took about 6 to 10 years. So it was much faster than we expected. So it did happen in our lifetime. And we're already seeing millions of people benefit from it in the clinics. Why would this be similar? And the reason is we can do a lot of the testing on embryos and embryo-like structures. And if we can do the scale up where we do it all in vitro, we don't do it inside of the animal, we could potentially do millions of embryos in parallel, which allows us to do a lot of parallel processing. The generation time, I mean, it could be, yes, they live 50 years, but they reach reproductive age at around 8 years for the females. And so so that's the time we're talking about. But also we can scale up by using this, again, highly parallel lab-based reproduction. We could get tens of thousands of these within the first couple of years. So I think all of these things conspire to make it possible. The other thing that was limiting for the first decade that we worked on this is we had almost no money. We had about $10,000 a year, which for anybody that runs a lab knows that that's not enough to run a project. And so most of the progress was made by volunteers, people that were in the Boston area biotech community, who would work like volunteer on weeknights and weekends. But that's no way to run a project either. So fortunately, after 10 years of that kind of starvation, we've now got hundreds of millions of dollars to work with. And I think that's going to help us get where we need to go on that project. I didn't raise that money. It kind of came in through Ben Lamb.
Still, we have the age reversal technology, hopefully, so you might see.
That's that too. Yes, that's the other reason we'll live to see it.
Fits together, I think, in this radical research. We have another question. It's about the stem cell science, stem cell. It's about the key challenges towards stem cell-based organisms in this de-extinction species conservation. Where do you see the challenges of species conservation, de-extinction, maybe population engineering in this stem cell technology business?
Yeah, well, at first it seems like stem cells play a small role. And in fact, with pigs, that whole engineering I talked about where we engineered 42 genes in the germline, that didn't really require laboratory in vitro stem cells at all. And of course, use stem cells inside the developing body, but that's you don't usually think of that as stem cell technology. But with elephants, I think, and for many of our ecosystem conservation efforts that will benefit because of this ability to study them at low risk to the endangered species outside of the body and in parallel and scaling. I think all of that will benefit from being able to make from pluripotent stem cells to make the reproductive organs and the reproductive cells that mature into gametes and then fertilize in vitro. In vitro fertilization is very well developed, both for animals and human, and so forth. So I think stem cells are playing a big role in our research so far on elephants, even though they didn't on the pigs.
We have another question that is more related to ethics and CRISPR-Cas. So in theory, it should be you. I'm going to read it. Katharina Wiesen wanted to ask which arguments are most important for you in the ethical discussion in the application of CRISPR-Cas, and which role a scientist plays in impacting on the lawmakers, and how to use methods on a daily basis in the future. She talks about modified plants or mechanisms to treat diseases. So CRISPR-Cas and the scientist.
Well, first of all, I would say it's not limited to CRISPR-Cas9. I think that there was already a revolution in play before CRISPR came along, which was based on recombinant DNA. It was based on protein engineering and so forth. But if we lump all these together as genome engineering, or broadly, including CRISPR-Cas9, I think I've mentioned some of the things that mainly influence me. One is the equitable distribution of technology and education about what the outcomes will be, both positive and negative. Having broad discussion where you don't just talk, but listen to all kinds of people who can think of how it could affect them positively or negatively. Now, in many technologies, there are winners and losers. If global warming, it will help some people, although it will hurt most people. And we need to, and so you can't just say this is going to do no harm, because almost every technology does some harm. Automobiles, for example, which are a net positive, you need them for ambulances and fire trucks and so forth. But they kill 1 million people a year. So that's a second thing that I worry about, is making that evaluation of winners and losers. And what you really are looking for, what I'm typically looking for, a win-win, where you really have almost no losers, and you take something that looked like was a trade-off and turn it into something which is positive. An example of that, merging the last two questions, was with stem cells. There was a great hue and cry. There were a lot of people that were very upset about using embryos as stem cells. And what happened is we eventually got better at using adults' cells as stem cells, and that became a win-win. So now we could get all the advantages of stem cells without using embryos necessarily. I mean, there still are uses for that, but the major point of contention was settled. And I could give many other examples where you get these win-wins, where you go from, sort of 50% are going to lose one way or another to more like, 99.9% are going to win.
Okay, I think we are fully smashed. It really excited. The active discussion on the chat was wonderful, and I very much enjoyed directly interacting with you and having the opportunity to do that. There's one question I know that is burning in the hearts of the students after that presentation, and that is there a way to join your lab transiently? I think that is something that I'm sure the master students will come up to me tomorrow and ask, hey, can we? And I would say, well, if you write a good application, there may be a chance. Is there anything you could tell them?
Yeah, I think that's true. Almost all of our graduate students go through the Harvard and MIT admissions process, which I'm not part of directly. So that's that. We do have students at earlier stages occasionally. We have visiting scholars, and we have virtual participation occasionally. And of course, I participate in iGEM and other worldwide events like this one to try to broaden as much as possible. But yeah, use your imagination. And there are lots of other good labs out there too.
That's true.
Very good.
Yeah.
Okay. I thank you very much for being here with us this afternoon in Germany and morning in the US. And I'm very much looking forward to hopefully meet you in the near future in person and exchange even more. Thank you very much. Also from the entire panel here, Jonathan.
Yes. Just like 80 is the new 60 in terms of aging, I think video conferencing is the new in person.
Hopefully not.
It almost feels like, but.
So thank you very much also from my side. And thank you to all of the audience that contributed to the chat and that gave us, shared their ideas with us.
Okay. Yes.
Thanks to all of you.
Thank you very much. Yeah.
Thank you. That's great.
Bye-bye.
It's wonderful to be back in Heidelberg. Thank you.
Yeah. When you're back in Heidelberg.
Well, I'm returning. I've been there a few times before. It's a lovely city. I miss it. Yeah.
Okay.
Hopefully again.
Take care.
Bye.
Bye-bye.